Hi guys, so this is the Macromolecules video. If you didn't watch the content video, it's okay. I'm going to be honest, this is going to be a really great video because I just did the whole thing. It didn't record. You guys know I'm terrible at technology, so this is round 2 for me and hopefully it's a little bit better for you. Now let's start with macromolecules. This is a big section for you. It's a lot to memorize. The actual information, not that difficult, but I do understand it's a lot, so let's try to get through it. Talking about macromolecules, these are organic substances. They're carbon-based. Not hugely important to your test, but good to know. But if you know anything at all, please, please, please know what the 4 macromolecule groups are. They are carbohydrates, lipids, proteins, nucleic acids. You have to know that. If you don't know that by now, you're struggling. Read your notes more often. All right, now let's start with carbohydrates. Carbs function as an energy source for us. It's a quick energy source. I talked about marathon runners using it. They carb load so that their body can tap into it easily and quickly. They also help with cell structure, but that's not hugely important. And then talking about the elements within it, so talking about the chemicals in it, it's carbon, hydrogen, oxygen. So from that Amoeba Sisters video that was kind of like the cheesy video, which I did put on the website, so you can watch it if you need that as a recap. That's a really great one. You can remember that acronym CHO, CHO, CHON, CHOMP, and just kind of help yourself remember it off the top of your head. But for this one, carbon, hydrogen, oxygen. Now, when we're talking about carbs, first we need to talk about how the classification breaks down. We talked about that monomers are our most basic subunit of macromolecules, or just any type of substance really, but a monomer is that basic subunit. For a carbohydrate, the most basic subunit is a simple sugar. We also know this is called a saccharide or a monosaccharide. You need to know all of the words for those things. Now, I refer to it as a Lego block to kind of help you out with it, but you have to know that it's a monosaccharide is your monomer, a sugar or a simple sugar. All of that is interchangeable for you at this point. Now, when you take 2 Legos or 2 monosaccharides and put them together, you get a disaccharide. This becomes a more complex carb. The more you add onto it, the more complex it gets. When it goes from being 2 to anything more than 2, now it's called a polysaccharide. I gave you examples for each one. The big one for your monosaccharides is glucose. You'll see it all the time throughout the year. It will never go away. Glucose will be around forever. You know it pops back up in cellular respiration and plenty of other places. Now, the disaccharide examples I gave you were table sugar, meaning like what you put in your coffee, your baked goods, or whatever, like actual sugar. And then lactose, which is the sugar in dairy products or milk. Now, remember that the OSE at the end usually indicates that it's sugar, so if you forget, OSE, those letters, OSE, are a good indicator that it is a sugar, and you need to know a sugar is a carbohydrate. Now, talking about our polysaccharides, the examples I gave you were glycogen, plant starches, cellulose is one of them. These are just really complex carbs. It takes longer for your body to break them down, which is why we recommend, okay, you should eat vegetables. It's because your body's working harder at breaking down these good quality complex carbs rather than just your simple sugars. Now, we're going to move forward and go into lipids, our second group. Now, lipids, I would say, is probably the most elaborate group. It's not super hard to understand. There's just lots of classifications within classification. So, to start off, there are 3 groups I need you to know within lipids. There's triglycerides, there's phospholipids, and there's steroids. Now, we know triglycerides are also called fats because we're lazy and we like to nickname everything. Triglycerides are fats, but you need to know what they're made up of. It's always made up of a glycerol group. We know that's the exact same. And then 3 fatty acid chains. Now, within this, we know you can have saturated or unsaturated triglycerides or fat. Talking about a saturated fat means that you have single bonds for carbon. So single bonded saturated fat is that the carbons within your fat are single bonded. They literally just have one bond in between them. Now, talking about carbon, we said that there are 4 total bonding sites for it to be stable. That means when it single bonds to each side, it has 2 open sides. Now it's going to fill it with hydrogen, so it becomes saturated with hydrogen ions. Now, for fats, the elements that it's made up of are carbon, hydrogen, and oxygen. The difference between carbs in this one is that it's mostly carbon and hydrogen. And when you look at a picture of a saturated or unsaturated fat, it's very clear that that's the case. Now, When we talk about an unsaturated fat, the only difference is it goes from being single bonded to double bonded carbon. The first time you see a double bond, it means it's an unsaturated fatty acid tail. Okay, so we said that on a glycerol, there's 3 of them. On the picture I gave you on your PowerPoint, you had the top 2 as saturated fats. The bottom one, once it has that one little double bonded carbon, it's now unsaturated fatty acid tail. You should have it on your notes somewhere or a vague picture of it. If not, go to the website, pull up the PowerPoint, and look at it. Now, outside that, there really isn't much to know on this one. You just need to know single bonded, saturated, what it's saturated with, and that this is for triglycerides. Our second group is our phospholipids. Now with phospholipids, it's really, really similar. You still have that glycerol group on there. You also have the 2 fatty acid chains, and then that third one that you have on triglycerides is now replaced with a phosphate group. But it forms a way different, ooh, my battery's dying, I'm gonna move you guys. It forms a way different structure. So we drew it and it kind of looked like our weird jellyfish situation. So what happens is that it's gonna die. We're good. So what happens is that you have this phospholipid group and it's hydrophilic. That means it likes water. And then you have the weird fatty acid tails hanging down off of it. You have 2 of them. And then you have your glycerol group in the center that kind of acts like the glue with the 2. Now, when it does this, you have your hydrophilic or the water-loving part, which is your phosphate head. And then your fatty acid tails are still going to be insoluble. So going back to biology, we know that fats don't dissolve in water. Any fats, they just don't do it because water's polar, fats are nonpolar, and they don't like each other. So your fatty acid tails are hydrophobic. They are scared of or don't like or are not going to break down in water. Now, I'm going to grab my other computer so I can see what's going on, but give me just a second. Oh, you can see Obi's fat self back there eating a bone. Disgusting. Not really. He's adorable too, but he's real fat. All right. Now, our last group within lipids is our steroid group. All I need you to know for steroids, it forms 4 connected rings of carbon. The example of it is cholesterol. And then what cholesterol does, it helps synthesize other steroids, it helps synthesize sex hormones. Other than that, it's in every single cell, so it's important to us. Other than that, I don't care what you know about steroids for this at this point. So let's move on and go to proteins. This is our 3rd group now. We're more than halfway done, thank goodness, because this is, ugh. So for our proteins, it has a wide variety of functions. Basically, it breaks down to, I want you to think about proteins as being what provides structure to our cells. So think about like a big, I don't know, I'm going to do this like weird hulky thing. A big bodybuilder is going to eat a lot of protein because they want to provide structure and hypertrophy and build up muscle in their bodies. So that's the way that I remember is that protein helps with structure. It also has other sources. So like it can be an energy source. It can help with hormones. It can bind with a carb and become a receptor. It can also function as an antibody. Okay, and if you want to go in and read about each one of those, you know that receptors are on cells and they kind of like look for stuff and pull it into the cell and signal for things. Now for your antibodies, it detects and destroys foreign substances. We talked about osmosis Jones a little bit with that. Now a protein's makeup is going to be your, what are we at? CHON. So we did CHON, CHON. Carbon, hydrogen, oxygen, nitrogen. Now, they can have sulfur too, not a big deal, just needed to know it because it's science and it's in there. Now, the building block for your proteins, meaning your Lego piece for your proteins, are amino acids. I feel like I have definitely tried to get this into everyone's brain at this point. Amino acid is your building block or your monomer of a protein. When you start to put those things together, then you get proteins, okay? So your most basic portion is an amino acid. Now, an amino acid is made of an amino group, an R group, and a carboxyl group. And remember, your carboxyl and your amino groups stay the same. The R group is what dictates what amino acids you have. You have about 20 amino acids in the body. You don't need to know what they are. You just need to know that they exist in your human body. Now, your protein structure levels, it just goes from primary to more advanced. So primary protein structures are just kind of like globular, and then as they get more advanced, they start to fold on each other, and we didn't really go that far into it, so don't worry about that for your test. Now, they do have two things on this slide that I'm looking at now. You have to know that the shape of a protein is called a conformation. This comes back when we're talking about enzymes anyways, but the shape of a protein is its conformation. When you have an alteration to that shape of the protein, it doesn't happen easily, but when you have damage to your protein, damage to the shape of your protein, or conformational change to your protein, this is denaturing. Okay, so damage to the protein is denaturing. Why this happens is for extenuating circumstances. This is things like change in acidity. So we'll talk about it more with enzymes, but when an enzyme in your stomach lives in an acidic environment, it thrives on it being in an acidic environment. Once it becomes basic, it has the potential to denature. Same with high temperature. If you have a fever, you know at a certain point you got to go to the hospital or you might die. And the reason for that is that your cells start shutting down on each other because your proteins start to denature. Huge, huge issue. So if your heat gets too high, if it gets outside of the range that it's acceptable, it can also denature and have a conformational change or a change to its shape and damage the protein so that it's no longer usable. Other ones that I've given examples of are radiation, electricity, and things like that. And an example I gave, like a real life example, was boiling an egg. It's the idea that like once you boil that egg, you can never go back to the weird, mushy, raw egg form. It's cooked, you've changed the protein, you've had conformational changes, and it doesn't matter what happens to it, it's never going back to being a raw egg again. Now, last part, get excited, we're almost done. Nucleic acids. This is our fourth and final groups for macromolecules. I am going to try to break it down as quickly as possible, but basically this is where we're talking about our genetic info. We said this is our part for DNA and RNA. Basically, they're the most complex, they're very large. They're made up of our last group, the CHOMP group, so CHO, CHON, CHOMP. So CHON, like Nancy, and P. So we have carbon, hydrogen, oxygen, nitrogen, and now phosphorus. Its monomer, or its Lego piece, is called a nucleotide. Pretty easy to remember because it's nucleic acid and now nucleotide. And when you put nucleotides together, you get these chains that then form nucleic acids. Now, a nucleotide consists of a sugar, a phosphate group, and a nitrogen base, essentially. The difference between RNA and DNA is what sugar you have. The two possibilities you have for this are ribose and deoxyribose. Okay, so for ribose, you have RNA. For deoxyribose, you have DNA. So it's the sugar group that changes within your nucleotide. And then you can piece all those together and create a strand of DNA or RNA, which creates a nucleic acid. Now, we did say there's two types, RNA and DNA, and this is talking about our genetic info again, so this is important to like mitosis and all that too. For RNA, it contains a ribose sugar group. It's a single polypeptide chain, and it folds into various shapes and it helps with gene control. That's all I want you to know about RNA. For DNA, it contains deoxyribose for its sugar group, and that's in its nucleotide. It's a double helix or a double chain, so basically what it looks like is two pieces of RNA have now connected to each other. The way they connect to each other is through a hydrogen bond. Now, hydrogen bonds are really, 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 really strong. Okay, so that's important to it, that it's a hydrogen bond that connects the two strands together, and it has a deoxyribose sugar instead. Now, for this one, it stores information within the DNA, and it's for coding proteins. The nitrogen helps code for it, and then we use it to synthesize proteins. And then our very last slide gives you a picture of it, but really you just need to know single strand, double strand, RNA, DNA, ribose, deoxyribose, and all of this is for nucleic acids, which have a monomer of a nucleotide. So at this point, take a second, recap, go back if you need it, look through your notes. This is everything you need, okay? Everything, and we got it done in under 15 minutes, not too bad.